welcome everyone to our today's session of Revolutionizing Healthcare. This is the second part of our empowering clinical trials with machine learning sessions. Today we will try to answer some of the problems we have pointed out during our first session. And we will start with a small presentation by Mihaela, who will summarize what we have talked about during the last session, all the different challenges that were posed last time, and put them together in a nice little field for us to work off afterwards, where we talk about opportunities with small conversations / presentations from Mihaela and other members of the roundtable today. And then in the end, all our participants will discuss together in a roundtable format. You can at any time ask questions in the chat or just mention that you have one. And during the Q&A after the panel discussion, we will then call you up. You can ask your questions, and I hope we have some time left to answer what you bring to the table. So during the last session, you can watch it on YouTube. You have the link here. You can later see in the recording as well, or just go into our YouTube channel to see the first session if you have not managed to do so yet. You can also read more about adaptive clinical trials on the Schaar Lab website, where you find more information and also more information on what we are currently working on, all our latest publications, and also a research pillar on individualized treatment effect inference. For our today's participants, we have thankfully gotten back everyone from last time. So excited to hear where their thoughts went during the past weeks about all the challenges we established. We have Professor Carsten Niemann, who is Associate Professor at Head of the Chronic Lymphocytic Leukemia Laboratory and Clinical Research Program for CLL at Rigshospitalet Department of Hematology in Copenhagen. Dr. Eoin McKinney, who's University Lecturer in Renal Medicine at the University of Cambridge, Honorary Consultant in Nephrology and Transplantation at the Cambridge University Hospital's NHS Foundation Trust. We have Dr. Pierre Marquette, who is Head of the Pharmacology, Toxicology, and Pharmacovigilance Department at the University Hospital of Limoges, Director of the U1248 Research Unit at the French Institute for Medical and Health Research, and President of the European Association of Clinical Pharmacology and Therapeutics. And last but not least, we have Professor Richard Peck, who is Honorary Professor in Pharmacology and Therapeutics at the University of Liverpool and former Global Head of Clinical Pharmacology at Roche. So welcome to all of our panelists, and you will hear from them later. And now we will start with a presentation by Mihaela, who will summarize what we've learned in the last session. Thank you very much.
Wonderful. So thank you very much, Andreas, and especially thank you very much to everyone who is joining us today for the first time. I would like to start by discussing briefly a summary of our discussion last time. But before doing that, I would like to start by highlighting the current situation that clinical trials are facing. We know, all of us, that they are difficult to run, they are expensive and ripe for disruption. I have summarized here the current situation by citing several numbers which I gathered from a variety of publications related to clinical trials run by pharma. The numbers that are associated with clinical trials run in academic environments may look somewhat better, but the situation still remains the same. You can see here, for instance, that the typical size of a phase 2 or phase 3 RCT trial has a large number of patients that is going to drop out. Also, you see high cost associated with running these trials. And the duration of running such trials is quite large as well. Also, I would like to highlight that the probability of success is relatively small as well, even for a phase 3 trial. So given the current situation, we believe that there are many opportunities to improve the current way in which clinical trials are run. And this is important since it has not changed in decades, despite major advances in machine learning and biostatistics. So what we are going to discuss today are innovations that we believe are possible to empower clinical trials to improve patient benefit, reduce the required patient numbers by replacing controls potentially and preventing dropouts, reducing cost per patient through improved recruitment, monitoring, and operations, improving the probability of success through better recruitment, predictions, trial design, and analytics, overall with the goal to reduce the cost and time savings by running better trials and improve the outcome of the patients, both within the trial as well as the drugs that are approved. So I have constructed together with my students the following table based on the discussions we have had last time with the various clinicians. And what you can see here is the four stages of clinical trials. And as you can see, we unravel from the discussion numerous challenges that are felt throughout the entire clinical development journey. For instance, in stage one, incorporating observational data, incorporating preclinical data, incorporating cross-trial results, running optimal designs of trials, as well as many others. And we are going to touch on a few of these points today. At the stage two, determining dosage, determining potentially personalized dosage, recruiting the right patients, retaining recruited patients, and many other opportunities. In stage three, at the analysis stage, trying to predict personalized response, doing improved subpopulation analysis, looking at competing risks, combining PK/PD models with other data to improve the analysis, as well as other solutions. And finally, even after the trial has been successful, improve the commercialization stage by making sure that the right treatment is provided to the right patient, by modeling disease progression, and by facilitating real-world data access and analysis. So what we have done in our lab following this discussion is map these different opportunities into machine learning areas that could attempt to address these opportunities and these challenges. So you see, for instance, that there are a variety of cutting-edge machine learning methods that one could envision developing to address these challenges. They range from causal machine learning to transfer learning, to inverse reinforcement learning and reinforcement learning, to time series analysis, to few-shot learning, and last but not least, a topic that we have discussed in a previous session, synthetic data. I'd like to highlight that this represents really cutting edge in machine learning that goes well beyond just risk prediction and risk prediction over time. So what is interesting and important is that technologies will need to be built to empower these different stages of the clinical trials. In our lab, we have developed a few solutions for several of these stages, and we will discuss a few of them today. But as it is going to become clear in this discussion, I believe that a lot more is possible, and we as a community, as a machine learning community, need to join forces with you, the clinical community, as well as the pharma community, to think creatively about how we can improve clinical trials with the overall goal of reducing cost and improving patient outcomes. Thank you.
Many thanks, Mihaela. What I'm going to do is just introduce some specific examples of clinical trial weaknesses, the sort of hidden problems that we don't always appreciate. And these aren't randomly selected. Many of the specific examples I'm giving are going to then be picked up by Mihaela in a couple of moments' time, and she'll illustrate very nicely how a recent method from her group can address each of these specific problems. So the way that randomized clinical trials, which of course remain the gold standard for evidence-based medicine, that's the foundation upon which current practice is made. But the way they work is by comparing group treatment effects. So we effectively stratify a cohort into two or more groups, and we allocate a treatment to those groups. We then look at the treatment effect, but we look at it in a group-wise basis rather than at an individual level. So what we're fundamentally doing is comparing grouped mean effects, which by themselves gloss over the fact that within each of those groups, there are a range of individual responses to that treatment, and the size of that group, the size of the group required to demonstrate that effect obviously depends on the variability between individuals in terms of their response as well as its magnitude. But the ideal really would be to dispense with the idea of grouped treatment effects and really understand instead for each individual, in other words, each dot on this plot, what impact the treatment is going to have, either to understand that after the study's been undertaken, but even better to understand that before the study's undertaken so that we can estimate who should actually be in that study in the first place. Because as we know, trials, RCTs that are set up to compare group mean effects, they're only restricted, often very heavily restricted by inclusion and exclusion criteria to one specific population in order to try and maximize the chances of unmasking this grouped effect. Consequently, they're very expensive. They often need a huge number of individuals, very time consuming, and in part that is manifest in their cost. And importantly, they only traditionally, and this changes a little bit, but only a little bit now, they only typically investigate a single intervention at a time. So we test one intervention in a group-wise fashion. So to put that in a concrete example, you can see the recent recovery trial in the context of the COVID pandemic. The overall effect that was seen in the context of the COVID trial, the recovery trial that was looking to randomize dexamethasone treatment in the context of COVID infection, did find an overall benefit of intervention. And you can see here that hidden within those group mean effects was a specific effect that was restricted really to those who had the most severe disease and who were on ventilators. And this is nothing new. This is an example of a pre-specified subgroup analysis within a clinical study. So you have to a priori have a suspicion of what subgroup might benefit most from your treatment, so you can specify that you're going to look in that subgroup. And this is a very modest step towards individual effects, but obviously it's only subgroups. They have to be pre-specified. What we really want to understand is, rather than these crude groups, what might be happening at the level of the actual individual. But the other thing that's quite striking about the curves, I'm going to stick with the recovery study, not because it's a bad study, but just because it's a recent study and a high profile one. The other thing that's striking about this is that you can see quite clearly from the curve that about 70 to 80% of individuals recruited into that study didn't meet the primary endpoint. They didn't have the relevant event. And consequently, the overall event rate is comparatively low. So we're recruiting people into this study, and well over half of them aren't having the event and aren't contributing to the endpoint. So they're accruing cost, they're accruing effort, and they're not really substantially contributing to our ability to find that endpoint. And if you add on top of that the fact that average dropout rates are around 20%, actually you're already losing a fifth of individuals before you even get that far. So trying to predict or understand the likely individual effects before we start a study can substantially improve our ability to find an endpoint and to see a treatment effect. Just to put that a little more firmly into context, this is a reasonably simplistic approach, but what it does is take a completed clinical trial from a few years ago, so a biologic treatment intervention for inflammatory bowel disease, and it looks at the power estimates on the basis of the observed treatment effect. And all it does on the y-axis to the left is show you the number of individuals that are required for that study in order to have sufficient power to find the treatment effect. On the right, you see the average cost per patient, and you can see how eye-wateringly expensive things become as you have to recruit more individuals. And on the x-axis, just going from left to right, you see a progressive increase in the baseline event rate. So if you can increase the baseline event rate in your study, in other words, if you can stratify and enrich and recruit people who are more likely to have the event in question, you can substantially reduce the number required in your study for the same statistical power to find the endpoint and drastically reduce the costs. even for comparatively modest increases in the baseline event rate. So I think understanding individual effects before the study starts and who should go into the study can have a huge impact in terms of cost and likely outcome of the study. This isn't something that's brand new. This is just a survey of existing studies, and those that use markers to enrich their populations have demonstrably have an improved ability to successfully transfer through each phase of the drug discovery pipeline. So from phase 1 right through to phase 4, and actually overall there's about a 3-fold increase in your likelihood of approval from early phase right the way through if you're stratifying your population in this way. So it's not just a more efficient, cheaper way of doing it, it's demonstrably a more cost-effective way of doing it, and you're more likely to be successful too. The other thing, as I mentioned, that clinical trials address traditionally, and this is accepting some of the more recent ones, but it hasn't changed much, is that they only test a single intervention at a single dose at a single time point with one duration of treatment. So in recovery, for example, people were given 10 days of dexamethasone treatment. The inclusion criteria specify when and how they're treated, but we don't really get an estimate of whether they could have been treated at a different time point in their clinical course. What would it have mattered if I had treated them earlier? Would it matter if I had treated them later? Would it matter if I had treated them for longer? And understanding that at an individual level is something, as you'll see in a moment, that may become possible through individual effect estimation. So the other thing that easily gets glossed over is the fact that we conduct clinical trials based on an included population, and our inclusion and exclusion criteria are typically almost comically restrictive. So we always get a clinical result. The RCT result then forms the basis of our clinical practice. That's evidence-based medicine. And yet the population we perform trials on is demonstrably not representative of the people we give the medicines to afterwards. So you can see on the right-hand side here, the trials that are just ranked in descending order of exclusion. In other words, what proportion of the population with that disease are excluded from a given study. This is the most comprehensive look I could find, but you can see that certainly half of trials are excluding almost 3/4 of patients with that disease. And yet where it's been systematically evaluated, we can see that the people who were ultimately treated with that intervention, almost 2/3 of them that are being treated would have been excluded from the original trial. So whilst we like to think that randomized controlled trials are a gold standard, whilst we like to think they're a firm and robust evidence base, clearly it's not the case, and that we perform trials in a small minority of people, then extrapolate it well beyond those confines, and that's arguably not appropriate. And that's, again, something that may be addressed by the methods that Mihaela will discuss in a second. And finally, one thing we assume when a clinical trial is undertaken is that the result that's identified is somewhat sacrosanct and will remain the case. So a clinical trial that was conducted 20, 30 years ago could become out of date. It could be that the magnitude of the effect that was seen then may not still be the case, given that comorbidities change, the demographic structure of the population changes, additional medications change, clinical practice changes. So once a result is established, we're pretty good at safety monitoring. We're good at surveying for adverse effects due to that medication, but there's no formal mechanism in place to continually monitor and reevaluate the efficacy of that intervention, despite the fact that we know clinical practice changes over time. And again, that is something that may be potentially addressed by some of the methods that Mihaela is going to raise now. So this was really a means of trying to introduce some of those key downfalls of our CT approach, our current gold standard approach, and now we'll hear about how they might be addressed by novel methods. Thanks, Mihaela.
Thank you. So as Eoin has mentioned, what we would like to understand is whether machine learning can use electronic healthcare records, so longitudinal data, to determine what treatments we should be using for a particular patient, given their unique characteristics. Now, this is a problem that is very challenging because it is not only a prediction problem, it is a causal effect inference problem, and it is a longitudinal one. So we really need to learn on the basis of both static patient variables, but also longitudinal patient observational data, both patient features as well as treatments over time, what particular treatment would be best for this particular patient. So on the basis of the patient history, which is biased, we would like to learn what will be the effect of a particular treatment and intervention over time. So we would like to estimate counterfactuals. You are seeing here a particular example, which is trying to use machine learning to estimate from observational data counterfactuals of different types of treatments over time, which will enable us to determine how to treat, when to treat, and when to stop treatments. The conventional way to deal with this problem comes from technology that was developed around 20 years ago based on marginal structural models, which represented the workhorse of biostatistics in this particular area. However, these methodologies have several disadvantages, inclusively the fact that they will lead to very high variance and bias if the number of patients is not large. So what we have done in recent times, you see here on top 2 papers highlighted in pink, were 2 methods that have been used to improve on marginal structural models using ideas from recurrent neural networks and sequence-to-sequence architectures to address this challenge. However, the most recent work in this particular area of estimating individualized treatment effects over time is using a neural control differential equation to do that. And the reason that I would like to highlight this most recent paper for 1 second is that it has the advantage that it can learn on the basis of irregularly sampled data, something that was very difficult to do before. So we learn on the basis of the observational data and the electronic health records, a continuous latent representation of the patient state. And then we are using this information to predict the outcomes in the future and do that for a variety of treatments at a variety of moments in time. However, an important disadvantage of this type of work is that it is of a black box nature. So in this very recent work, we showed that this significantly outperforms both machine learning methods as well as biostatistics methods. But this comes at the expense of a method that is black box in nature. So what I would like to do, I would like to highlight out of the entire work that we have done in this particular area, and you can take a look at our research pillar dedicated to estimating individualized treatment effects from observational data. I'd like to highlight one particular work, which is interpretable. And the setting in which this work has taken place is a setting where we have an electronic health record, which we know is a longitudinal collection of records that contain repeated measurements of a patient's health condition, which is captured at irregular time intervals. And although the treatments may also vary over time, many studies consider a point treatment where the treatment allocation is performed at some observed time and stays fixed after this time during the study. So a limitation of what I'm going to show to you next is that we will only enable point treatments. However, this is often the case if the treatment is, for instance, a surgical operation, like for instance in the transplant case or other surgeries, or in settings where we have long-term medication for chronic diseases where the treatments do not change frequently. We refer to this setting that I'm going to discuss as longitudinal and irregularly sampled data with point treatment setting, or for short LIP. And in this LIP setting, again, I'd like to highlight the fact that this is different from the conventional setting in causal inference because we observe the pre-treatment outcome over time, leading to the treatment allocation. And these treatment outcomes may unveil the inherent temporal structure in the outcome time series, leading to better individualized treatment effect estimation, personalized treatment effect estimation. And as you see in there, these yellow dots that you see are highlighting the potential counterfactual outcomes that we would like to estimate. And the shaded area that's shown to the right represents the point after the treatment has been allocated. So we would like to estimate for the particular individual, given their unique characteristics and outcomes prior to the treatment, what would be the effect of giving a treatment or not at this particular moment in time. And for that, we have developed a methodology that we call SyncTwin, which learns a patient-specific time constant representation from the pre-treatment observations. And SyncTwin issues these counterfactual predictions of a target patient by constructing a synthetic twin that closely matches the target in representation. And the reliability of the estimated treatment effect can be assessed by comparing the observed and the synthetic pre-treatment outcomes. Most medical experts can then interpret the estimate by examining the most important contributing individuals to the synthetic twin. Let me show to you how this looks like. Again, unlike conventional methods, whether coming from biostatistics or machine learning, synthetic twin has the advantage that it does not use the learned latent factor to directly predict the outcomes. Instead, it uses these variables to find the contributors and what patients have contributed to this, and the importance weights, such that we can construct a synthetic twin. And this brings two bonus features. Both are important to medical applications, these two features. The first one, which is highlighted here, is that we can calculate an individual estimation error bound based on the actual and the synthetic outcomes before treatment. Hence, a clinician can accept the recommendation in terms of treatment only when the error bound threshold is below a certain desired level, and reject it otherwise. So we can have hardcore confidence estimates associated with the predictions in synthetic twin. A second important feature of synthetic twin is that we can identify the most important contributors to an estimate as the one who received the highest weights. And hence a clinician can examine these contributors, these patients, and whether they are indeed similar to the target individual according to them, and validate and interpret the estimate. So you see here, for instance, that synthetic twin can identify the most important contributors to the estimates. How does SyncTwin work? Just very briefly, I would like to highlight the 3 steps involved. So first, the temporal covariates are encoded as representation vectors. Then in the second step, the synthetic twin of a treated target individual is constructed as this weighted average of the few contributors to the control group. And in the third step of synthetic twin, the difference between the observed outcome and the synthetic twin outcome estimates the individualized treatment effect for this particular individual. The ultimate test came for this particular method when we try to reproduce existing RCT results by using observational data. And for that, we have tried to use the observational data from electronic health records in the UK called CPRD, and try to unravel the results and reproduce the results of a prior clinical trial, which was this heart protection study that estimated that the statins LDL's lowering effect was reduced with an average treatment effect of minus 1.26 millimole per liter in the first year follow-up. And the question that we wanted to address is whether SyncTwin, if applied on the basis of observational data, can lead to the same outcome as this prior clinical trial data. And the answer is yes. when we are using the CPRD data to see if we could have indeed identified a similar effect, we saw that SyncTwin is able to estimate the average treatment effect to be indeed to be 125 milliliter per mole. And indeed, this is very different than current baseline methods, which are failing to reproduce this type of findings. And now back to you.
Thanks very much, Mihaela. A couple of points that I would stress there from how that fits into the context I was describing before, because I personally find it absolutely fascinating that from a health record data set, the individual effects are able to be firstly estimated sufficiently accurately that we can reproduce the same effect that was seen in the comparable clinical study. But of course, that's not the major point of SyncTwin. That's trying to reproduce the group mean effect. The major point is that it's able then to estimate the effect for every individual within that cohort that it's estimating the group mean of. So it's able to do so much more than that. And what Mihaela described about being able to estimate not just the impact of an intervention, but the timing of the intervention, I think extends this well beyond what clinical trials can currently do. Because I can't think of a clinical trial that is asked the question of when a treatment should stop. That's not a question that's commonly asked. But by asking whether something that's currently there would lose its effect is something that this can broach. Also, the fact that we know many of the treatments are extrapolated well beyond their founding clinical evidence base means that we can effectively in silico test in that additional extrapolated population whether the treatment effect is comparable. In other words, whether it's appropriate to have extended practice into that population that extends outside of the clinical evidence from the original study. And what you see in the slide at the moment are just relating some of the aspects that SyncTwin can address back to the original projection that Mihaela outlined. So the stages here aren't the phases of clinical trial design. They're different stages that you would go through if you were planning or undertaking a clinical study. So at the planning stage, obviously, SyncTwin can incorporate observational data. It can estimate individual effects for a previous intervention or an intervention that's similar to the one you're considering making in your clinical trial, allow you to identify which individuals might be most appropriate and consequently recruit better. It helps us to incorporate results from prior trials, learn from them and apply those to new studies. Of course, in theory, you could estimate individual effects for different doses of the same intervention and consequently get an idea of what dosage should be used in a subsequent study with regards to conduct. Identifying good subpopulations in the right patients. Well, if you can estimate an individual treatment effect across your entire population, then you can quite easily start to identify clusters who are responding better and who would be the right subpopulations to recruit into your study. And obviously, as that model is interpretable, you can not only identify those subpopulations without having to suspect them a priori or define them a priori before your study, but you can understand what are the traits of the individuals that are responding better, so we can start to learn more about the disease we're treating at the same time. Of course, the individual effects are actually founded on being able to predict a personalized response, and doing subpopulation analysis afterwards becomes much more feasible as well. As I mentioned at the end of the introduction, we don't really have a means for monitoring efficacy of interventions after trials have been done. We're very good at monitoring safety, but by applying this to electronic health record data, as Mihaela's outlined, it becomes feasible to monitor efficacy through electronic health record data and to see whether group mean effects for an intervention are comparable to what we would have expected from the founding studies that led to them being used in clinical practice in the first place. And we can actually monitor clinical practice over time and make sure that we're doing the thing that we think we're doing and having the effect we think we are. So I think there are broad ranging applications of even this one approach into the way we conduct trials at the moment. So Mihaela, Thanks very much. I think we're going to pass across to Richard now.
So Mihaela and I will tag team the next few slides. This and really just looking at another component, perhaps, of getting to this point of estimating the individualized treatment effect. This slide conceptually is something you've seen before, the idea of electronic health record data being the source data from machine learning to help guide treatment choice. I just put it up there to introduce two, well, in one case, remind everyone of one concept, but introduce one new one. So I hope in the future, we're not only doing this on electronic health record data, I hope we're actually doing this on data within clinical trials. Because when you think about developing a new drug, which is most of my background, you don't have any real world data. You don't have electronic health record data. You only have clinical trial data. And yet I hope we can still learn a lot more from that. And on the right-hand side of this slide, I'd like to bring people back to something that has been mentioned before, but maybe I can emphasize it a little, which is when you're thinking about the choice of treatment, typically we default to which drug. Maybe we might think about which combination of drugs for some diseases, but we should also be thinking about what dose for the individual patient. You know, that's not how many drugs, most drugs really are used today. They're all used at a fixed dose, and that's probably not the best way to use some of them. And if we really want to improve both trials and how drugs are used, that's something to pay attention to. And I think I hand back to you now, Mihaela, to talk a little bit more about some other methods.
Yes, so thank you so much, Richard. So what I really wanted to really highlight here is this idea of looking at estimating individualized treatment effects for multiple treatments. This is, of course, a very challenging task due to the need for an individual's potential outcomes to be learned from a variety of data, in the case of electronic health records, biased data, without having access to counterfactuals. So it's really estimating these different counterfactuals that is challenging. And this is especially the case when we need to estimate the effect of multiple treatments, where multiple counterfactuals need to be determined. What we have done a few years back is to build a method on the basis of the well-known by now in machine learning generative adversarial networks, or for short GANs. I'm sure that many of you already have heard about GANs because they have been used recently to generate art or synthesize music. Also, GANs were used to age faces of photographs of people or make them younger, or to show how an individual's appearance might change given different types of conditions. So this is, if you like, a way of doing counterfactuals that you have seen in the popular media. On a more serious note, the GANs have been used to detect glaucomatous images, helping for early diagnosis. But we are using here GANs in a very new way. We aim them to estimate these counterfactuals and estimate individualized treatment effects for a patient. And this is why we call our framework GANite, so GAN plus IT. And we generate proxies of the counterfactual outcomes using a counterfactual generator, and then pass this proxy to an IT generator in order to train it. And by modeling both of these using the GAN framework, we are able to infer based on the factual data while still accounting for the unseen counterfactuals. Now, while much attention has been given to the problem of estimating the effect of individualized treatment effects on the basis of discrete interventions, so in the previous example, GANite was looking at no treatment, radiotherapy, or chemotherapy. As Richard has mentioned, even more challenging is to look at continuous valued interventions, such as treatments associated with a dosage parameter. This was even more challenging for machine learning because now we really need to estimate from data the effects of such continuous value interventions. And we tackled these problems a few years back by building on a modification of the GAN framework. We call this framework SciGAN, and this is a flexible solution that is capable of simultaneously estimating the counterfactual outcomes for several different continuous interventions for different dosage. And to address the challenges associated with this continuous interventions, we really needed to develop new machine learning methods for our discriminator. And what we did is we built a hierarchical discriminator that leverages the structure of the continuous intervention setting. I will not bother you with the details, but you can take a look if you are interested. Instead, what I'm going to do is I'm going to turn back to Richard to introduce the next setting.
Thanks, Mihaela. So Eoin introduced or reminded everyone of the recovery trial and in particular the finding of the value of dexamethasone in ventilated patients with COVID-19. And I guess one of the questions we were interested in or are interested in is defining the patients who benefit as the ventilated ones good enough? Is that granular enough? Can we get more precise in identifying which patients should receive dexamethasone? And that probably will have something to do with the level of immune response in those patients. If you have a patient who's got a very active immune system, perhaps dexamethasone is helpful, but actually maybe some patients whose immune system is already relatively depressed and dexamethasone may actually be harmful. And building on that, why would every patient need the same dose of dexamethasone? That's the way it's used now because the trial only looked at 6 milligrams and we only have that population level data. But when we get to the individuals, maybe not everyone needs 6 milligrams. Maybe some people need more. I know there are trials ongoing looking at larger doses, but they're still in populations. And we were trying to think at the individual level. So if we can go to the next slide, please, Mihaela. We're not starting from no knowledge. We already know quite a bit about both the drug and the system. And I think one of the questions to consider is how do you incorporate that data? We know something about the relationship of dexamethasone efficacy to activity to its exposures, the PK/PD. I'll come back to that in a bit more detail on the next slide. And we know quite a bit about the immune system. And you can build a model of a systems-based model of the different components of the immune system. And indeed, that's been done and published. The first version, I think, included about 10 cytokines and 10 different cell populations within both the adaptive and innate immune response systems, and is really quite a complex model. But you can simplify it down to something that looks a bit like the cartoon on the bottom of this slide, where you can just show the relationships between the innate immune response, the adaptive immune response, the viral load, and the drug, and how they interact on each other. And those can all be parameterized and linked with ordinary differential equations. It's the sort of thing that clinical pharmacologists do fairly routinely. There are some issues, though. Many of the variables that you might measure are certainly not available in the clinic and are often not even available to highly detailed research labs because they're very difficult to get hold of. And what you have, though, are just the observed clinical variables. So what you're measuring is not necessarily what you incorporate in the model. So if we can go now to the next slide, thanks, Mihaela. These PK/PD models are developed in the research labs. You've got the linking of the PK, the pharmacokinetics, the drug concentration, to the PD, the pharmacodynamics, the drug response, often a pharmacological one. And you may know quite a bit about that. But that's not what the clinical side is really interested in. What the clinicians, of course, are interested in is, well, what happens to the patient? And we can link PK to that, but we're often not very good at linking the PK to the pharmacological and physiological variables to the clinical outcomes. And we have further problems, which is, actually in something like recovery and in many of the trials that are done outside pharma, you can't do PK/PD at all because you don't have concentration data. You don't measure the drug concentrations. Even if you did, actually, and maybe heretically to some of my clinical pharmacology colleagues, it might not help very much because variability in the drug concentration may not be actually what matters. It may well be variability in the patient's state that's actually much more important. You typically don't model the biomarker changes and outcomes, and you're modeling a population. PK/PD is about populations again. It's not useful for making individual predictions. So what can we do? And if we go to the next slide, can machine learning help? And of course, the answer we propose will be yes, but it's not probably the sort of conventional machine learning, because as we all know, it needs a lot of data. And in clinical trials, you often don't have a lot of patients. You may know a lot about them, but you don't have a lot of patients. And as we've heard already, and as we all know, the output is a black box, which is very difficult to then know how to use. But there may be a way forward to managing some of these problems and meeting this interest that I certainly have of trying to do machine learning or incorporate machine learning into the clinical trials of drug development. And so with that, I hand back to Mihaela to wrap this up.
Yes, so thank you, Richard, for that. So inspired by this idea, we proposed a novel hybrid model that embeds a given pharmacological model, such that the PKPD model highlighted by Richard, into a larger latent variable machine learning model. In this particular case is a system of neural ODEs. So what we have built is this latent hybridization model, or for short LHM, that marries the pharmacology models with the machine learning models, thereby creating this hybrid model that is both more efficient than existing machine learning models, but also, importantly, more interpretable. We note that in this LHM, we use observational data to learn both the evolution of the unobservable latent variables and the relationship between the measurements and all the latent variables. This includes the expert variables from the pharmacological model and the latent variables of the larger machine learning model. Just a few observations. This LHM model is much more powerful than the well-known solutions developed in machine learning. And also, this would reduce to a pure latent neural ODE model if we would omit the pharmacological variables. However, we do not want to do that because, as Richard has mentioned, this would amount to discarding prior expert information from pharmacology, which is evidently undesirable. And also, as noted before, our approach is driven by the idea of incorporating this prior expert information from pharmacology into a machine learning model. The advantages of that are that now this enables us, this LHM model enables us to unravel the link between the expert variables and the clinical measurements, thereby bridging the gap between research and the clinic. Secondly, the inferred expert variables from the clinical data provide additional insights to clinicians. And finally, from a prediction performance point of view, we do not need so much data as a machine learning model that does not incorporate such expert knowledge would require. Also, we note that practical extensions of such LHM will include static covariates. So this will enable personalization to a particular characteristics of the patient. And you can find information about that in our paper. But also importantly, we made one more extension, which I think is important. As Richard has mentioned, often the pharmacological model may not be correct. This may be wrong in multiple ways. The obvious way is that the functional form might be misspecified. For instance, a linear model might be specified when the truth is actually nonlinear. And many existing techniques can address such misspecification, and they can be integrated into the LHM model. And I have highlighted that in the paper. Just to say one word, what is interesting is we have compared this LHM model against a variety of machine learning models which are uninterpretable. And what we can see is that this model is not only providing the interpretability, but also is able to outperform the existing machine learning models that are uninterpretable because it injects within it these inductive priors, these inductive biases that are coming from the pharmacological model. So we have the benefit of both interpretability, but also higher efficacy from learning from limited data, which is important in machine learning. Richard, back to you now.
Clearly, the machine learning idea is complex. It's, it's something that's still going to have to be presented. But when we think about the clinical decision support, I mean, and I think I can wrap up the whole session here, what we're moving towards is something that is capable of giving individual predictions. I'm not sure whether we've got the slide that actually shows the kind of output that we have. Yeah, thank you. And this is the kind of thing that the existing setup does. And as I said, it's a crude model. We actually know it's wrong. We know it's incomplete, but it's able to begin to give predictions on what to expect in the patient, and to incorporate and to do that in a way that is hopefully meaningful for the clinicians, such that you can use this information to be guiding what you think you need to do next. I think maybe 1 minute or 30 seconds on wrapping up. I mean, what we would hope to do now would be to begin to identify, what does good look like? What do you need to see happening in these output parameters for a patient to indicate this patient is on the road to recovery, whereas this one's getting worse, then you need to make some different interventions, and to provide some guidance on what and when, to Owen's point about the timing of intervention, those additional interventions would come, but do it in a manner that is meaningful and usable by clinicians.
I wanted now to start to open the discussion and invite Pierre and Karsten to join us. And I know that Eoin had some questions for them in this regard.
Yeah, thanks very much, Mihaela. I wanted to start just by asking Pierre and Karsten, and please fill questions in the chat if others have them too. Ask them about implementation of the sort of approaches we've been discussing, and where they might see this fitting best, and how they might see these approaches fitting best into clinical practice, because it's one thing to demonstrate what is possible and another to actually impact it into practice. Karsten, I think you had a particular study in mind to raise in that respect.
Yeah, thank you, Eoin, and thank you for really nice presentations. And as you know, I have a background as a clinician and working half-time research, half-time with patients. And I always sit in front of my patients, wonder how can I actually speed up the implementation of data-driven, personalized treatment approaches, as you just demonstrated very nicely here. And what I would like to bring up is the paper that I just shared in the chat, where rather than randomizing individual patients and waiting for the process of informed consent and strict in- and exclusion criteria that we know from standard clinical trials, whether we can actually use the approach with a cluster randomized trial, where you randomize between the different treatment sites, the different hospitals, and implement your decision support tools with the guidance based on these very nice models that you're presented here, and have the clinician use this in periods of times and not use it in other periods. And what is demonstrated here for pancreatic cancer, it's absolutely outside my expertise, is that they actually significantly improved the outcome for patients. So I just wonder if this is actually the path forward to implementing and simultaneously testing algorithms and decision support.
Many thanks, Karsten. I also thought that was a really innovative way of trying to bring an algorithm in. And I guess for me, there are two aspects to this. One is whether you can use ML approaches to improve care almost as the intervention. But the second is whether the trial itself can be informed and changed by ML approaches, which is, this is sort of trying to straddle both of those in some ways. Pierre, did you have anything to bring on that?
Well, yes, I would say the SyncTwin system or algorithm might be very useful to try and run personalized or precision medicine clinical trials, because in this way that would be possible to tailor the amount of drug or the dose or the nature of the drug that is used based on the predicted outcome or the counterfactual outcome of the patient. And that would be a very good way of selecting patients, because this is really a problem we have when we run clinical trials for individualized interventions. We don't know how to select outliers or the patients that would benefit most of this type of intervention, because we are using drugs that have been proven efficient in this population. So we expect an average effect. And what we want to bring in is an additional effect to those patients that would respond poorly. And we don't know how to identify them. So that's really a nice way of doing that. And I think it might also be used in clinical trials, in, I would say, RCTs, just to figure out which patients respond poorly and why. And this may help designing better trials in the future, either for the rest of drug development or for other indications, for instance. So far, we are not very good at that. We only use biomarkers, but biomarkers are limited. And so trying to find out who are the poor responders might also be a very good addition to traditional RCTs.
It's probably worth also saying there that what we've talked about so far are treatments that are already in existence. So there is training instances to learn from. But what you're talking about is really learning from instances that we have and extrapolating that onto new therapy. So actually knowing about how we use current treatments can still be used for novel trial design too, which I think is an important point.
Yeah, that was 2 different applications possible. Yeah.
Oh, and I think just very quickly, I would maybe add one thing, which is, I now know of at least one example where a phase 3 trial was done as part of a development program, incorporating an individual treatment arm in addition to a fixed treatment arm. And that's a drug, I'll put it in the chat, but that's a drug called faricimab, which Roche recently had approved in the US. I don't think it's approved yet in the UK or Europe, for macular degeneration and diabetic macular edema, where the individualization is how frequently you give it, because it's an intravitreal injection. You want to give them as infrequently as possible. And that didn't require machine learning. It was a much simpler algorithm, decision tree, but it was included in the phase 3 pivotal trials.
But it's probably a condition where you can easily check out the pharmacodynamics.
Absolutely, but
which is not the case.
The precedent. I agree, that's not usually the case, but it sets the precedent that if you know how to measure the pharmacodynamics, you can incorporate individualized treatment arms into phase 3, even from a pre-approved development program.
It's a step in the right direction. Yeah. Mihaela, I can see that very encouragingly, our chat is filling up with questions.
So maybe we should take questions from the audience. I see a first question from Timing Liu. Timing?
Hi, thanks for the great conversation. So I have a question for Dr. I was wondering, so like for the ITE estimation, we normally require some treatment that has been recorded in the electronic health record. So how are you able to estimate the ITE if we do not have the like if it's a first trial and we do not have the treatment effect in the recorded?
Yeah, I think it's a great point. I guess it relates to what Pierre and I were talking about a second ago. If you don't have the training data, can you learn? Well, actually, I think from there, we know that treatments are not complete islands unto themselves. If you're going into a particular population where interventions have failed before, that may still be useful prior information, what treatments have succeeded, and help you stratify your trial accordingly. And obviously similar prior treatments and the patient population in which that's worked could still be informative. I don't know if there's a more formal way that could be approached. Mihaela, what do you think?
And I wonder also whether we could estimate on the basis of the available data which patient would most benefit from having a new drug, and hence enrolling those patients in those trials, such that we make sure that the patients would most benefit from an alternative medication, or maybe we know on the basis of the observational data that they will not so much benefit from the existing drugs, and hence the promise of a new drug would be most efficient. Maybe that's something additional that one could do in terms of using machine learning to determine who should be enrolled.
Yeah. May I ask a follow-up question on that? After that last.
Maybe there is somebody else. I apologize, but Dr. Quan Chu Yang has a question as well. Would you like to unmute
Sorry,
yourself?
can you hear me?
Yes.
Yes.
Yes, we can hear you.
Okay, thanks for every master and professor. I'm Kwang Chu Yang from Taiwan, Kaohsiung, Municipal Mission Hospital. And my professionalism is cancer and big data research. After hearing the talk of every master, and I'm wondering, can we use the lab to do some cancer research, such as genomics or pharmacomics or immunomics to predict drug use in real world from clinical trials, or even design a clinical, maybe we call it virtual clinical trial, to see what the effect of drug, what is the effect of drug. Okay, thank you.
I don't mind. I mean, I'll take a start, but I'm sure there are others who can join in. The answer has to be yes, but I think there are some restrictions. I mean, I say yes because it's been done. When you think about the EGFR antagonists, they were originally, at least one of them was originally used in the general population until it became apparent that it was only working in the KRAS wild type, I think it was. And that was, the label was retrospectively changed because the information became available from the genotype, the tumor genotype, to guide which treatment was actually working or which patients the treatment was working. And then there are similar examples for drug-associated adverse events. I think the problem is how easy that will be to do at the individual patient level. Once you, because if it's a relatively simple test that you discover predicts response, okay, perhaps that can be developed and made available. But if it's a complicated set of tests that take a long time to do or that are not widely available, that is going to be more challenging. But it's perhaps the sort of thing that it's part of the patient baseline characteristics, isn't it, that get incorporated into any subsequent predictions.
I think.
Thank you, Richard. Okay.
I'd just like to comment on that it also means that we have to deal with a number of health authority regulations if we implement this kind of drug testing and other companion diagnostics. So it's definitely a path we should follow, and we're currently running a EU-funded project on that, combining biomarkers from the lab with data. But there is a number of different regulatory perspectives that we need to address for this, and I think it's really important translational path to join what we can do from a data perspective and from a lab perspective, and demonstrate the path towards implementing it into clinical practice.
Okay, thank you, Carsten.
Perhaps to come to your question about immunomics and genomic data, I mean, it's obviously quite feasible to identify potential interventions that can modulate genomic pathways. And if there are drugs in existence, so we're really talking about repurposing place here, if there are drugs in existence that modulate those pathways, then this can be a mechanism by which those could be rapidly estimated because you have training data for them. Now, the interesting question becomes whether you have training data on which that drug has been used in, let's say, cancer. If it's being repurposed from another context, you're looking at a transfer situation. If you have sufficient data where its use for another indication happens to overlap with cancer, then you may be able to estimate that. But I'd be interested, it would be interesting to, as a thought experiment, to wonder whether you might be able to get sufficiently powerful estimate of the individual effect in a completely separate context, and what the overlap would have to be to allow you to do that.
If you allow me, I'm going to finalize this discussion with a question to all of you, the clinicians that are online. I wanted to ask you, for us, the machine learning community, if it is to consider these different stages and the different challenges that we highlighted, what do you think, what is your guidance to us, the machine learning community? What shall we look at first? What do you think are the main problems that you think we should consider as the most important problems to tackle. Could I go one by one to each one of you? Maybe I start with you, Pierre, and then I move on.
I would start by the end, but it's because I'm mostly involved in personalized medicine, precision medicine, so I'm trying to improve patient outcome with existing treatments. So obviously I'm very much interested in stage 4. And then I would say stage 3 is so challenging in terms of number of patients, money involved, etc. So that there is a very high challenge here. So I would go backwards and say stage 4, then 3, then 2, and then 1, probably.
Karsten, what about you? What is your recommendation? And are there specific topics that we should pay attention to?
So I think I would go for your stage 3, predicting personalized response, and then combining it with the optimal design of trials. So essentially challenging your synthetic twin approach to make it able to predict also time to event and contrafactual outcome, and put that into an optimal trial design where we can, on an ongoing matter probably, test whether it actually improves the outcome for our patients and identify the patient subpopulations where it really improves the outcome.
Thank you very much. Eoin, what is your recommendation? Thanks.
For me, I think the greatest impact should come from stage 4, because that's where we have most data to learn from. But perhaps that's being too simplistic, because I think perhaps we could do better than that and join stage 4 back up to stage 1, because stage 1 is where it's hardest. So if we can learn more about where we have most data, but then implement that through informing how to do the new interventions better, I think that would be perhaps the coolest thing.
Thank you. Richard?
Yeah, the issue with going last is, of course, everyone's already said the things you were going to say. And you know from previous conversations, Mihaela, that I strongly support Owen's point about joining 4 up to 1. It's a loop. It's not just a linear thing that then stops. But I would also jump on the predicting personalized response. Recognizing to start with, that's going to be with real-world data on drugs that are available and that are used widely. But if we really want to change how things get done in the future, we also need to be looking at how to adapt those methods so they can be deployed on the smaller numbers of patients that are included in drug development. Because otherwise, we're always going to be chasing our tails. With every drug that comes up, we'll always be in that starting already handicapped, because we don't know, we've got something that's coming in with a fixed label, and we're not able to change that.
Maybe final question. I know we are running over time, but of course, coming up with new ideas and new methods is wonderful, but obtaining regulatory approval is something yet different. Do you have any final thoughts on that, any of you? And then maybe we can wrap it up.
Frankly, I think it's critical to actually drive uptake. Otherwise, you can do the greatest science in the world, it won't get used. I can certainly quote examples of drugs where some beautiful machine learning has been done to understand how to improve the dosing. It doesn't get used because it's not in the label.
I think for me, Mihaela, I would put it out a question to everyone here and probably to regulators as well. If it's possible to demonstrate, and it hasn't quite been done yet, you've shown it for cholesterol lowering effect. If you can show clinical outcomes can be accurately estimated at an individual level on EHR data. In other words, you can do trials, you can repeat trials in silico. Surely that evidence on extended subpopulations that were never in the trial, that should be admissible, right? That makes perfect logical sense. But I would be interested to see if the regulators would take it.
But I think actually, maybe the regulators are not the most, the hardest challenge. I think actually the pharma companies, and convincing them that they should put as much efforts and resources into developing these approaches as they do to developing new drugs. Because I'm really fascinated being in the area of malignancies and hematological malignancies to see the speed and the resources put into developing new treatment molecules and identifying broad groups to treat with those. But if we put the same resources into demonstrating that identifying subgroups and treating those and adjusting the treatment, then I believe we can very fast develop the needed documentation and evidence for this.
A few years ago, Mihaela, I would say that's almost impossible, but things have changed because now the regulatory agencies are accepting or releasing drugs with only phase 1 or phase 2 evidence, which means that if we can bring more for these early access drugs, more evidence based on the digital twin or SyncTwin technology or anything else, that would be a plus and not a minus for these regulatory agencies. So I think there is really an opportunity here, and I would begin with this type of application and drugs.
Thank you very much for the panelists and everyone who joined us today for all the thoughts and ideas. Thank you very, very much.
Yeah, thank you very much from my side as well. And I'm surely can say the same for the audience. That was really, really interesting. And I'm looking forward to learn more about this. If you want to learn more about this, we will put this session up on YouTube as soon as possible, so you can rewatch it if you like, or share with your colleagues, hopefully. The next session will be in September, so we're taking a small summer break to relax our brains a little bit, and make sure to share the sign-up link with your colleagues if they would like to sign up for our mailing list, where we will put out all the information once this session is on the video and when we start our next session as well. And yeah, thank you very much for coming, and I hope you have a lovely summer.